Please join me in welcoming to the Distinctive Voices podium, Dr. Andrew Endy.
Thank you very much. Thanks so much. Thank you for joining tonight to celebrate science and to support the National Academies. It is an incredibly deep honor for me to be here. It's quite humbling to be associated with scientists of such accomplishment as Dr. Benzer and his friend, Dr. Brenner. I was told to have some fun with you tonight as well, and I intend to do that. And I hope you're hungry for science. I hope to leave you with some things that will seem strange, that you might not agree with, and I might be wrong. And I hope you'll find my mistakes. So we've got a full plate, and I've set this up sort of as a three-course meal. Our appetizers are going to be aliens, then we're going to talk about computers, and then close with the bioeconomy. And we'll see how it goes. Okay? All right. So these three topics are meant to introduce synthetic biology to you, and I presume many people haven't heard those words put together before. And this first topic, our appetizer, aliens, is meant to introduce you to the idea that we can learn about biology by synthesizing things, by building things. So that's what I want to try and get across quickly. One of the questions in biology is how come we're here? Where did we come from? Genesis is a model. The RNA world is a model. We arose from spontaneous chemical interactions. Turns out I hadn't heard about another model, and it's shown here in this paper. It's called directed panspermia. I read this paper just very recently. This version is written by Francis Crick of the DNA structure fame and Leslie Orgel. Directed panspermia is the idea that life arrived on Earth from somewhere else. It was sent here. Well, why don't we read the paper together? Here's figure 1 from the paper, and they represent on this y-axis time in billions of years. Imagining going back to the beginning of the universe, over 10 billion years ago. And then they say, okay, well, at some point, our solar system comes about, there's Earth and life on Earth, and that turns out to only be, of order 4 billion years ago, which means there's 10 billion years before that for all sorts of other stuff to happen. Like life could have started somewhere else, and then it got here. And what's very, very interesting is there's so much time available for passage, for transit, for transportation of life from one place to another, billions and billions of years. They note that this is so long, you don't need to be going very fast to go quite a long distance. And that's what they work up in this interesting paper. They propose there could be a spaceship that is transmitting life between planets. Now, it's not in their first-order model going to be transmitting people. It's going to be transmitting microbes, bacteria, because they're going to be easier to ship. And then they note that, if you only went 60,000 miles an hour, which we could get to with rockets, but you moved at that velocity for 1 million years, you cover quite a bit of distance. Is there any evidence for this? Now, we see movies, that talk about this, but how about reality? Well, I started following up on this and was surprised to see a paper that was published 2 years later, roughly. And it has the title, Is Bacteriophage, Phi-X174 DNA, a Message from an Extraterrestrial Intelligence? Question mark. Well, bacteriophage, Phi-X174, what is this? Bacteriophage are viruses of bacteria. And it turns out that this particular one, Phi-X174, it was the first organism with a DNA genome that human beings sequenced. Fred Sanger and his team sequenced this in 1977, read out its genome. And within 2 years of that, there's a paper in the literature that says, is the genome of this bacteriophage a message from aliens? One of the interesting things about phage, these bacteriophage, is they are probably the most abundant, the most numerous reproducing biological systems on our planet. It's estimated by the ecologists now that there are 10 to the 31st, 10 followed by 31 zeros of these reproducing phage particles all over Earth. Millions per milliliter in the surface waters, for example. Well, that's kind of interesting because if you wanted to send a message and you wanted people to find it, one of the smart things you'd do is you'd make sure the message was everywhere to be found, as opposed to hiding it deep in a cave someplace that nobody would ever go look. You'd put it in some of the most abundant things out there. That's one good reason to think about looking for a message in a phage. But it turns out there's more, and it comes from the reading out of this organism's, this phage's genome. So this is a very old schematic from when the genome was first sequenced. All the letters were read out, and it's showing in a circular form what the genome is thought to look like. And all those boxes you see from 9 o'clock to midnight, there's a box with the letter A inside it. That is a region of the genome that has a gene that encodes a protein that's associated with the virus doing its business. Now, what's very curious, right, so that's one gene. Gene A is associated with copying the DNA. What's very curious, though, about Phi X is that inside gene A is another gene. See that other box between 11:30 and midnight? There's a gene inside a gene. Gene B has nothing to do, first-order replication of DNA. It's a scaffold protein that helps assemble the particle. Well, how's that come about? Earlier, people had thought one gene, one enzyme. And now we've got a piece of DNA that's got multiple genes making different proteins. It gets better. If you look at the detailed letters for gene B and you count the number of blocks of letters, it turns out that gene B is 121 blocks long. 121 is an interesting number. It's 11 times 11, the square of a prime number. Huh. How would evolution ever select for something like this? If I was going to put a message in, I'd start to do things that were, well, not expected, and then use prime numbers, and that's what these researchers caught on to. So they decided to decode this sequence of DNA. They imagined all the different coding schemes, pretending they were the aliens. And then they made patterns with what they found. And that's figure 3 in their paper. So you have an 11 by 11 matrix, and they're trying to decode all the blocks of DNA using different binary coding schemes. And you can see the patterns they found. Does anybody see the message? They concluded that there was not a message. But raises a question. Maybe they're looking at it the wrong way. It raises lots of questions, right? But it also raises a question, what could you make? Could you code something in that wasn't just about the virus? Well, it turns out the answer to that one's yes. For example, a first-year undergraduate up at Stanford looked at a matrix like that and said, hey, what if I could encode the Stanford logo? No, an S with a tree. I've got 4 bases of DNA. I can use 4 colors. And, white, red, green, and so on. And I could turn that into a binary code, and then I could turn that into a DNA code. And then I could lay out the sequence of DNA that encodes my pattern. And then we can synthesize that DNA. And now we're storing that pattern in a strand, a molecule of genetic material. Now, if you're not a first-year undergraduate, but you're a Harvard professor, you can write a book, it turns out. And you can turn your book into a digital bit stream. And you can put that digital bit stream into a DNA synthesizer, which I'll tell you more about later. And you can print the DNA encoding your book. So now your book isn't a physical object that's really big. It's a little speck of white salt that has billions and billions and billions of copies of your book. That's pretty interesting if you're a librarian. Yeah. Okay. Well, what does this have to do, though, about Phi X and science? Let me try and come back to that quickly. I'm showing you the genome again, the sequence of DNA for the entire organism, up top, now in a line. And you can see, for example, that B is inside A. They're overlapping. And, well, does it matter? We didn't find the alien messages, so should we care about this at all? What would happen if we re-architected the genome? Could we decompress everything? Could we pull B out of A and give it its own spot? Could we make what's shown just below? Certainly we can design it by reorganizing the sequences on a computer, and then we can build it. We can synthesize it. And we can take that piece of DNA and see what it does. And we did that. Paul on my team did that last year. It turns out when we did that, we couldn't find any big difference between the performance, the infectivity of the natural virus and the one where we had ripped all the genes apart and decompressed it. What does that mean? Well, it means that we don't know what the importance of the overlap is, at least right away. But we keep going. And this is where it gets really, really interesting. Remember how that circular plot seemed so simple? It just had about 11 boxes on it? But as we read and read and read and read and read and read and read more examples from nature of genomes of this type, we see that they're all slightly different. And when we use our computers to analyze those sequences, we find that, yes, there are those 11 genes. But then we find about 100 other hidden genes. And that's what some of which are shown here in all these colors around the rings and rings and rings. And we don't think they do anything, but we're not quite sure. Are they hidden messages? Are they evolving genes waiting to show up? Right? When the environment changes, we just don't know. Well, it turns out what we can do with synthetic biology is we can start to do something about it in the laboratory. We can say, instead of all this complexity we don't understand and just looking at it, how about we try and erase it? All of it, and see what happens. So we took Phi-X174, and we erased all these hidden genes by making hundreds and hundreds of changes, and we called it Kleenex174, which is our synthetic design. And we built that by synthesizing the DNA. So the first thing I want to try and communicate about synthetic biology is, as humans, we're very good about learning by taking things apart. You want to learn how a car works? Take it apart. Have all the pieces on your driveway and in your yard. And you'll get some understanding of the car. But if you really want to learn how a car works, you're then going to take all those pieces and try and put it back together. Right? And which pieces can get rid of. Right? So synthesis, construction, a way of tinkering is a very powerful complementary approach to learning, to discovery, to science. That's what synthesis, synthetic biology, brings to the table. When we've done this, and this is now unpublished work, which we're happy to share, it just barely works. Hardly grows at all. We think we've disrupted some cryptic protein that the geneticists, using previous approaches, hadn't found yet. And we don't know. And we're sort of in the middle of that mess right now, where we're trying to use the approach of synthesizing and building and combining that with tearing apart and seeing why things are happening to figure out what has been missed previously. Okay? Okay, on to the entree. The reason I put computers here is I'd like you to struggle with me to appreciate how early we are in partnering with biology, not as a science, but as a material which leads to novel technology. So I'll give you ways of thinking about this. From this photograph, which I'd love to go, I have no idea where this is, but I'd like to go there. I'd like you to imagine, looking at the materials you see depicted here, what material would you use to make a computer? The sand, because it's got silicon, and computers are made of silicon. Perfect. Now, the first thing I want to emphasize is if you tried to take the sand and build a computer, you wouldn't just scoop it up and pour it on the table and work with it with your hands and expect to get a computer. Even if you don't do this work yourself, you would know that there's probably a very sophisticated process of refining and purifying and reworking the materials to make the semiconductors needed to implement electrical computers, right, that we're familiar with. We don't merely take the raw materials from nature and repurpose them, and expect them to work right away. So think about all the steps. Even if you don't know it, just imagine how many steps must be involved from taking those natural materials and making a refined object like a computing machine we're familiar with. I want you to, nobody's done this yet. We're just beginning to dig into this. I want you to imagine the equivalent process that we're starting to build, starting to dive into with living matter. I want you to think about reworking living matter to such an extent that we can make new things, really new things. I don't know what that means, okay? But I know that we can really refine these materials, and that's what I want to give you a hint of. Now, it turns out the computers we build, we don't build them from the silicon in my lab. We build them from the biology. And in particular, we build them from the biology that you can't see, not the palm tree, but it turns out from the bits and pieces of the bacteriophage we were just talking about. The bacteriophage that have, I don't know, 1 million or so friends per milliliter of seawater. And we're going to take bits and pieces of those natural biological systems and begin to repurpose them to make new types of computers. And I mean to give this to you as an example of what it will mean as we struggle to figure out how to refine and make new stuff using living matter. I want to show you this movie. This is an example of bacteria reproducing and growing. This is a movie we take on the microscope, and it's a time-lapse movie. The movie runs for about, 3 hours of real time, compressed to run much more quickly. And those black blobs that are going like this and then boom and pinching, those are E. coli, the common bacteria E. coli. Now, to an engineer, as an aside, the most amazing thing about E. coli is that it's going like this and copying itself. It's a collection of tens of billions of atoms that in a period of 24 minutes can make another collection of tens of billions of atoms. That is an incredible technology, which we can't come close to as engineers with any of the other stuff we do. So that's the real miracle of life and the challenge to engineers thinking about biology. Now, there's something else happening in this movie. You can see that a few of the E. coli turn green. And the E. coli that turn green have been infected with a bacteriophage that we've engineered to take a protein from a jellyfish that's green. All right, so you can see when cells are infected. Does that make sense? All right. Now, there are 2 cells that get infected at the beginning of the movie. The movie's looping. In the top right, you can see a cell gets infected. Right there, turns green, and it pops, goes away. Here's a close-up of what we think that looks like. What's happening is that infection leads to replication of the virus particle. The cell bursts open or lyses, and that releases more virus particles into the environment to carry on infections in other cells. So that's why that cell is going away. In the bottom, you see another cell that turns green, and it doesn't pop. That's because the bacteriophage that's infecting this cell can do 2 things when it infects. It can pop the cell and release more progeny, or it can hide out and go dormant and integrate its genome into the genome of the host until it decides, if you allow me that language, to come out later. So implied in this natural system, this is going to come back to computing in a moment, implied in this natural system is the existence of a switch that can control whether or not the infected cell will burst, releasing progeny, or result in a dormant infection, and the virus is hiding out. I know what to make with switches, all sorts of things, if only I can get my hands on it. Now, the people whose hands are really getting on this are the 2 gentlemen shown here. Jerome Bonnet from Montpellier, France, was a postdoc at Stanford for the last 5 years, and he's just gone back to France to start up his new lab. And his colleague, Pakpoon Subsutorn, or Tone, from Thailand, is a PhD student at Stanford. And these are the 2 gentlemen who have implemented data storage and logic using bits and pieces of bacterial viruses. And that's what I want to give you a sense of. So how would you store data inside a cell? There's so many ways to answer that question. I can't spend time on it. But I just want to acknowledge there's many, many ways we can imagine engineering data storage inside cells. Why do I want to store data inside cells? To keep track of data. I might want to keep track of how many times a cell has divided so that I can figure out how old it is. It can just tell me how old it is. Maybe I'd like to reprogram what it does as it gets older. Maybe I'd like to program how cells differentiate to form a tissue if I need to regrow a tissue or something like that. Right? Maybe I want to record an environmental signal, like a pollutant. Have I been exposed to this? Excuse me?
Record virus attacks.
Could record virus attacks, yeah. Lots of reasons. So I don't want to get hung up on that. The way Jerome decided to store data is shown here. This is the sort of Apple incorporated abstracted version. We have a strand of DNA, and we can engineer a sequence on that strand of DNA to be recognized by the enzyme that normally inserts the virus genome into the host genome. But we're going to change the sites using work going back to 1982 so that instead of an insertion reaction, we get a flip. In other words, we're going to try and do this. We're going to take a piece of DNA that we engineer and cause it to be flipped. So DNA is like a railroad track. It has two strands. And you can imagine a turntable flipping from one orientation to the other. That's what we're doing. If we can flip it in one direction, maybe we could flip it back. And we have two directions the tracks could be running. Well, two directions, kind of interesting. That means there's two states. That means it's binary. That means we can get to digital data storage. For example, if it's pointing to the left, this section of DNA, we're going to call that a 1. And maybe we'll make the cells turn red in this example. And if we flip it back and it's pointing to the right, we'll call that a 0. And maybe we'll make the cells turn green. Again, we're going to use the enzymes from the bacterial viruses, known as integrases, to mediate this flipping. So if we make one of the enzymes, we'll flip. If we make two of the enzymes, we'll flip back. That's the idea. Can we do it? Yes. So here's the experimental data. We start with cells, and the little data register is pointing to the right, so the cells are green. And we set the bit, and we flip the DNA, and the cells become red. And we remove that signal, and the cells stay red. They're storing the information. And then we flip the DNA back, and the cells are green again. And we remove that signal, and most of the cells stay green. You see that one little cell turn back red? It's not perfect yet. Right? The reset reaction is a little bit messy still. And what's neat about this is we can flip it back and forth, back and forth, back and forth, back and forth, back and forth as many times as we want. This is the first rewritable digital data element inside a genome. And this technical word at the top, non-volatile, means that once you write the data, you don't require any energy, any cellular resources to keep the data, which is very important if you want the cell to keep your information around and not have it require a lot of energy and get selected against during evolution. Any case. So we did that. We've got 1 bit of data storage. Now we have 2 bits of data storage. I literally have a 2-bit data storage system, which is kind of embarrassing. But I want to keep going. What else could you make with DNA digital switches? This is the puzzle we happily found ourselves dealing with. We can flip DNA back and forth. What else could you make with that? And here's where it got really interesting, because we hadn't really understood what we did at the time we got that flipping working. And I found myself going back and reading for the first time the work of this gentleman, George Boole, who published his work around 1854, if I remember correctly. His most famous work is probably this book, The Laws of Thought. And if you haven't read it before, here's one of my favorite parts. He's talking about how we find it useful to understand our universe, our world, our life experiences by using language and particular words within our language. For example, we use conjunctions, and or, Mr. Boole writes. Trees and minerals. Right? Barren mountains or fertile vales. It's simply handy to talk about the world we experience by keeping track of things using words like and, or, and so on. So handy have we found this to be in our experiences that in any material in which we can do it, we have implemented the equivalent of Mr. Boole's observations. It's called logic, Boolean logic, after Mr. Boole. We implement the equivalent of an and operation, an or operation, and so on. And I show you this because a lot of times when people think about a computer, I and others make the mistake of thinking about the computers we know. You know, the computer in this thing, or what I'm using for the presentation. But computers aren't necessarily in silicon. Right? It's wherever we make them, however they work, in wherever we make them. And I missed that. That's a really key insight. And the reason it's key is because if you just think we're trying to make biology like electronics, then you get into all sorts of problems appropriately. But if you think we're trying to make biology like biology, then you can do it. So let me give you an example of that. Remember, we got really good just a moment ago at flipping DNA. We're going to use that to make a different type of computing element. The element we're flipping in this case has this T symbol, and it's basically a check valve. It allows for a flow in one direction only. So when the T is in this orientation, it's going to block the flow of something. Well, what is the something? The something is the reading out of DNA. So the wire here is not an electrical wire, it's a piece of DNA, and what we're blocking is the reading out, the enzyme that flows along DNA and reads out genes. Enzyme's called RNA polymerase, and it drives this process of transcription that causes DNA to be read out. By having the T in this configuration, we block transcription. We block the flux or current of RNA polymerase. However, if I flip the DNA, the check valve is backwards, and the current for reading out DNA can pass from left to right. And if I return the check valve to its original state, I'm going to block the current again. All leveraging this idea of reusing a digital genetic switch. Flipping DNA back and forth, I can now control flux or current of reading out DNA. This is what we call the transcriptor. It's similar to a transistor, but it's not a transistor. It's very different from a transistor. It's a single molecule digital switch, perfectly digital switch, because the DNA can only be in one of two states. All right. Sometimes it turns out that transcriptor is like a transistor when you go to use it. For example, if we want to implement, meaning make, the equivalent of Mr. Boole's and, right, mountains and trees, what we do is we take two transcriptors and we put them in a row, in series. And that's just like you would do with transistors and electronics. So for example, in this configuration, no current passes through. No reading out of DNA to the right. If I flip the first transcriptor and I flip the second transcriptor with two different control signals, then I allow the current to pass left to right.
Are those module 3?
We'll take questions at the end. Yeah. Here's a different thing we can do, and this is very different from electronics. Instead of flipping two transcriptors, we can make a more complicated object in which the flipping sites are nested within themselves. So this is going to be called an exclusive OR gate. The signal to the right is only high, only happens if the control signals are different from each other. They can't be the same. They can't both be low or both be high. So in this configuration, nothing passes through. If the black and white sites, oh, by the way, these little triangles represent bits and pieces of bacteriophage. Okay? If these black and white triangles are activated, we turn things upside down, and things flow. If the blue and orange bacteriophage sites are recognized, we flip the whole package again, and it blocks. So I can only have one of the signals, not both, in order to get read-through. This turns out to be very different than electronics, where you can't control one object using multiple signals at exactly one point on a wire. Okay. All right, I know that's a little bit esoteric, so let me push on and zoom out and we come back to it. We looked at the major operations that Mr. Boole likes in his logic family, shown on the left, and we came up with different configurations of DNA to implement each one, and we predicted how they should behave, and then we made them, and it worked. We got all the major logic operations working. All right? Now, the enzyme that does the flipping is called an integrase. This is the enzyme that normally in nature integrates DNA from the virus into the host, but we're using the integrase to flip the DNA. And we're using this flipping operation to implement Boolean logic. So I had a hacker working in the laboratory, and he came up with a nice name for this. He said, you should call this Boolean integrase logic Gates, or Bill Gates. And that's what we did. I want to just show you another aspect of this, which is pretty neat. On the left, you see a movie of cells growing. In the middle, you see the level of the signal needed to flip the gate. And on the right, you see the level of the signal that comes out of the gate. And you'll notice that the color on the right is much more intense. And basically what this is demonstrating for the first time is that we can make what's called an amplifier. You know, if you're playing a guitar, it's very quiet, but if you amplify it, it'll be very loud. That's very important in computing because it allows you to pass information further along and to restore information if it begins to degrade. You could think about telegraphs needing relay stations to transmit a signal a very long distance. And so one of the hidden technical significances of our architecture based on flipping DNA is we have this amplification baked into the system. Okay, but can we do anything useful with this? I am the wrong person to ask. And to give you a hint that the answer is yes, I have to refer to the work of some colleagues. And I'll try and do this very briefly. It's Weston Whitaker's work in Justin Sonnenberg's lab. I didn't know these folks, but they saw what we had done and said, we could use that for something. They decided, you see, they study how the microbes work in our gut. And they want to try and figure out how that works. So if you have colitis or something going wrong, you know, how would you begin to think about dealing with that in a modern way? And the problem with this is it's very hard to study microbes inside mammals, as you can imagine. And the classical technique oftentimes involves using mice and then sacrificing the mice and collecting tissue samples from the gut and staining it and then looking at it under the microscope and trying to figure out which bacteria are where and what are they doing. And what these folks realized was they could take our ability to flip DNA and all of a sudden use that as a genetic tape recorder that would integrate signals inside the gut itself. The microbes would become the recording instrument to study themselves. So for example, they could have a signal based on the state of the immune system, a signal based on, say, whether or not the microbe has seen fructose. And if both the fructose sugar and the immune signal is there, A and B flip, right? And that results in a flipping of the DNA, and that DNA is in the bacteria, and that bacteria might come out of the mouse. And you can sequence it. You can read it out, and you don't have to kill the mouse. It's a new type of recording. Instead of a piece of hardware, like we see around all this building, all these beautiful instruments, we can have genetic measurement technology. That's what they've done. Okay. One last bit about a computer, and then I'll move on to dessert. Monica just graduated with her PhD. Monica Ortiz, shown here, she's now doing a postdoc at Harvard. And Monica decided she wanted to program cells to talk to each other. Right? We can store data, we can implement logic. Another part of a computer is communication, sending information from one place to another. Now in biology, those places are probably oftentimes different cells. Right? We have tissues and whatnot that are quite complicated, and that depends on communication. And what Monica figured out how to do was take a different bacteriophage and hijack it so that the bacteriophage, instead of transmitting its DNA back and forth, would transmit whatever message Monica wanted to send. So she built a new communication channel for sending information based on DNA, where you can package up any piece of DNA you want into a virus particle. You now have a transvestite virus particle containing your DNA. And when that virus particle infects another cell, it delivers your message. It doesn't result in a reinfection. It just results in whatever your message says to do. Does that make sense? So here's a simple example. She takes cells which are broadcasting two different messages. On the left, green. On the right, red. And the cells start swimming toward each other. And you can see they're going to collide in the middle of this plate. And where they collide, there should be exchange of messages. And if those messages are transmitted, you should begin to see yellow cells showing up, the combination of green and red. You can just begin to see in this slide about 30% of the cells are yellow. Or here, much more yellow. All right. So if you can send DNA messages back and forth, what messages would you like to send? I don't know. One of the things Monica tried was sending the Bill Gates back and forth. Oh, sure, we can send computing elements back and forth to program a population of cells to implement behavior, not just one cell at a time. That's going to become very interesting later when we want to program patterns and growth of particular shapes. All right, on to dessert. And I'll try and do this quickly so we have time for questions. Dessert here is going to be this thing called the bioeconomy, whatever that is. And so let me present it quickly. Genetic engineering turns out to be about as old as I am. And if you look at the products that are on the market domestically, they add up to about $300 billion a year of domestic revenue. And that turns out to be 2% of our economy. 2% of our economy is about as much as mining, extracting stuff from the crust of the earth. This is for a technology which is a generation old. What does it break down to? Medicines, like recombinant production of insulin for treating diabetes, foods, oftentimes controversial, genetically modified crops, and stuff. I'll come to that to give you an example of stuff, because it seems kind of odd. Also very interesting is historically, this is growing at 10 to 15% a year. Don't know if it'll keep growing, but that's a pretty impressive clip. You can read more about this in the bioeconomy blueprint that the White House put out last year, or Rob Carlson's analysis, and so on. Let me give you a quick taste of stuff. It turns out in laundry detergent, there's an engineered enzyme called subtolysine. This is a depiction of what the structure of that enzyme looks like. The enzyme was engineered back in 1980 to work at cold water wash temperatures. This is the enzyme that degrades other proteins. So if I get spaghetti sauce or something on my clothing, I need a little molecular Pac-Man to clean that up. Right? The significance of getting this enzyme to work at cold water wash temperatures is you don't have to necessarily heat up your laundry water. And if everybody were to do that, we could save 10% of our domestic hot water heating bill, which is about 100,000 barrels of oil a day in the US. That's quite a lot of oil. That's about 20x the Gulf of Mexico oil spill. That's stuff, right? So you begin to see why making things, partnering with the engineering of biology already has had these hidden impacts. But I want to hit you with this. Most of biotechnology hasn't been imagined yet. Now you might be thinking, great, because I don't want it, right? But I'm not sure. I think we need it. So let me give you some things you might puzzle over. Here's a suspension bridge. I'm a civil engineer by training. I worked for Amtrak one summer. I like big stuff. I like to build things. I like suspension bridges. Wouldn't you know it? People have grown a double-decker suspension bridge. Takes a long time. It's passed down from one generation to the next. It fixes carbon as it grows and gets stronger. Huh. Here's a different project. This is reprogramming E. coli, which are normally this dull brown color, to be like a box of Crayola. E. coli on the left are programmed to make a red pigment. E. coli on the right are programmed to make a purple pigment. What would you do with bacteria that can change colors? I'm not the right person to ask. The students who did this project went to the Royal College of Art in London and asked the designers, what would you want to bring to market? The designers said, we'd like to take these pigment generators and put them in yogurt and also add in sensors to different disease states of the gut, the alimentary canal. And depending on what disease state you have, the bacteria will change color, and what comes out of you will indicate the medical specialist you should go see. They called this the so-called scatologue. Now, right next to that word, you see they also put the numbers 2049. They thought that we would not be ready for this technology. Not that the technology wouldn't be ready, that we would not be ready for such technology for several more decades. It's interesting, though, because medicine, as we well know, is limited by our ability to get information into and out from patients. And a biological technology that allows us to get information out from people at very low cost is quite relevant. Most people can't afford access to the technologies that go in to get the information out. Could we do this? Could we imagine a new type of biotechnology that is based on making living matter fully engineerable? And the way I think about this is to look backwards and say, over the last 70 years, as engineers, we have gotten collectively very, very good at engineering information systems. What should we aspire to get good at over the next 70 years? And for me, it's programming living matter. Not to make things crazy, but to reinvent how things work so they work better. It's not obvious that we can do this. It's not obvious that we can take this type of material and make it fully engineerable, tens of billions of atoms at a time. And the reasons for that has to do with the basic science of biology. It's very different from other types of materials we know how to engineer. It's very noisy at the atomic level. These things copy themselves. They evolve. They're very diverse. We all know this. These present huge new engineering challenges. Oh, by the way, there's another challenge associated with this. If you actually make any progress whatsoever, almost guaranteed somebody's going to freak out. Maybe we shouldn't be doing it for valid reasons. Part of synthetic biology has been to wrestle with this question. And I'll just point you to, for example, a study we did in 2003, where we asked the question, could we do it? And if so, what would we start with? How would we make biology easier to engineer? And if you read what we published a decade ago, we said you should do three things. You should make standard biological parts that can be reused. You should separate design from construction. And you should try and struggle to make this thing called abstraction work, which is hard to explain in words. I have to show you. In other words, what we were trying to do was figure out how to take the core of the engineering process, design, build, test, redesign, build, test, redesign, build, test, until you get something working and make it go faster, better, more times in parallel. So we could just learn how to engineer the stuff. One thing we highlighted was this technology called DNA synthesis. I've alluded to it. Let me brief you on it quickly. In these bottles are chemicals which are the four bases of DNA. A, T, C, G. They're in a form that allows for new molecules of DNA to be constructed from scratch. You connect these bottles to a machine called a DNA synthesizer. You've heard of a musical synthesizer that makes music. A DNA synthesizer makes DNA. You feed into the keyboard, if you will, the information, the sequence of DNA you want printed, and the synthesizer prints the DNA. You then have to take the DNA and put it in a cell to get it to do anything, or to store your book, or whatever it is you want. I'm very biased. I consider the synthesis of DNA to be a technology that is the most important technology of our time. It's at least as important as silicon wafer manufacturing. It's got to be more important, because it's the material that operates life. Nothing against silicon. Silicon's really important. This is probably more important. It allows you to go from abstract information to physical genetic material. Here is a depiction of why it's important in a way that will hopefully allow you to think of how come it's important in ways I haven't thought of yet. On the top is how biology works historically in nature, and for most of the time, almost everywhere. We go from one generation to the next via direct descent. I've got a 9-month-old boy at home. We didn't make him in the lab, right? But what synthetic biology is allowing us to explore is another path. We can take a generation, a point in a lineage, and decode the information using sequencing, reading out the DNA. And now that information is on a computer system, on a network. We can change it as information. Then, by resynthesizing the material, we can put that back and start a different lineage. In other words, we can break the chain of direct descent. This is because sequencing and synthesis of DNA, reading and writing of DNA, are advancing to a point where genetic material and genetic information are interconvertible or fungible. This is a big deal for more reasons than I can imagine. I'll leave you just with some ways of thinking about it. Remember when entertainment used to be in the form of vinyl platters? Well, it's kind of hard to copy that. But then we had digital CDs. It was kind of hard to copy that, but you could do it. But then you had digital media files over the internet. And CDs are kind of going away. Well, that represents that entertainment media and entertainment information become interconvertible. Right? Or how about another one, which isn't one I like too much? Smallpox is locked up to keep us all safe from smallpox. It's at the CDC. But the sequence for the smallpox genome's on the internet. Huh. You see where this goes? Okay. That's synthesis and why you should begin to wonder about it. Let me do abstraction very quickly. Remember I mentioned briefly I want these data storage things to make counters, count cell division, track aging, and so on. Well, here's an example of an 8-bit counter. See all those flipping things? Each one is 1 bit, and if I could line them up like this, I could count up to 2 to the 8th, or 256. That's quite a lot of counting inside a cell. Cells often don't divide that many times. How would I ever possibly make this? You know, that thing I showed you before, where we made 1 bit of data storage, it turns out that took us 3 years. And 750 design attempts, different DNA sequences we had to engineer, build, and test just to get that one flipping thing working back and forth. Well, we've got to do better than that, and this is where abstraction comes in. If I want to make at the top something like an 8-bit counter, I can't spend all my time working down at the level of DNA. I have to be able to call down and say, hey, could you give me some bits? And then I just put them together, they work. And then the person working on the 1-bit systems will call down and go, hey, I need some pieces of DNA that work. Right? And you put them together, they work. That's the idea of a standard part that works. And then somebody would say, hey, I actually need the DNA. And the person down at the bottom knows how to make the DNA. But the person at the top who's working on the 8-bit counter, the tumor-destroying bacteria, whatever system is you want, might not need to know that DNA is made up of 4 bases, because that information's hidden down at the bottom. It's much like we use our computers to send information around, but most of us don't know how to program computers, myself included, using zeros and ones. We use very powerful, high functions, if you will. Could we implement something like that for engineering biology? This has been very controversial, very technically controversial. There's no such thing as a standard biological part in biology because things will always behave differently when you change their context, and so on. I'm simply here to report today, over the last year, after 10 years of work, and I won't go through the details of this, we've shown that it's not impossible to make standard biological parts that are reliably reusable across changing contexts. We haven't done it yet for all the types of genetic words or functions we want to compose, but we've solved the problem for cases that are the worst case scenario examples. The significance of that is this abstraction idea isn't off the table. Remember I talked about the logic elements. Every one of those DNA designs worked the first time. It didn't take us 3 years to do that project. It took us 3 months to do that project and 3 months to publish the paper. We'd never had that experience before. It was because we had black boxed these digital switches, the flipping of the DNA, and we used available standard biological parts that didn't exist until about a year ago. Why does this matter? Well, if you go to Segovia and you see this aqueduct, you know it matters because standards, like the standard rock shown here, allow for coordination of labor. Somebody can make the rock, somebody can stack the rock up. There's another reason it matters, particularly so in biology, and it has to do with this gentleman, Bill Joy, who's a computer scientist, but also famous apparently for observing the following. No matter who you might be, most of the smartest people work for somebody else. And this turns out to really be true. I only have a few PhD students, and we're okay, but most of the smart people don't work for me. Most of the biologists who discover the gizmos we need and we repurpose, well, that's not us. Right? That's Graham Hatfull in Pittsburgh or Maggie Smith in the United Kingdom. So how do I figure out how to coordinate better with all of my colleagues? My future colleagues, it turns out. So we started making standard biological parts and giving them away for free and enabling students to make things like those colored bacteria. And they started doing it. And when they did that, they would then give their parts back to us so that they could be shared with next year's students. And we did it again and again and again. We've been doing this over the last 10 years. And the number of students has gone up. Thousands of students each year now participate in what's called iGEM, the International Genetically Engineered Machines Competition. At the beginning of the summer, they're given a collection of thousands of free-to-use BioBrick parts. At the end of the summer, if they make new parts, sometimes they give them back so that other people can use them next year for free. The collection grows. This is important if we want to get to a future where you look at all the things our civilization needs and you unpack just one of those boxes and say, how do we reinvent the manufacturing of everything we use, now derived from fossil sources, and build it with biology? We don't have enough people who can do that. We don't have enough technology. We need to scale. We need to make real Joy's Law. We need to make new capacities. This is quite literally the construction site where I work. This will be the first time next year when this building finishes that I will work in a bioengineering building. Okay? It's not big enough. We're not thinking big enough. This is what I want to leave you with. So for example, I have a pine tree in my front yard in Menlo Park with my family. And it makes these objects every year. They're amazing, these pine cones. They grow from apparently nothing, well, really the atmosphere and the water. Right? And what do I do with this stuff? They're big. They're bigger than these bottles, 1,000 of them. And they grow undifferentiated balls of sap, and then they fall off. And they get swept up and thrown away. And I, as an engineer, cannot ride my bicycle every day back and forth and ignore this anymore. This is a state-of-the-art, self-compiling nanotechnology with solar panels that grow themselves. And I throw it out. It's crazy. And it turns out in the town of Menlo Park, to the best of my knowledge, we do this to the tune of 500 pounds per person per year. 16 million pounds of matter compiled from the atmosphere that we throw away or compost. What can I make with 16 million pounds of matter? Well, it turns out the world's microprocessor supply is only about 1 million grams a year. A couple million pounds. All the silicon chips for the next year. I can make 8 times as much of that with the matter compilers of Menlo Park. How do I reprogram my pine tree to grow computer chips? I don't know. I've been working on it. I went to this wonderful farm in Monterey, Far West Fungi. They're the major supplier of organic mushrooms in San Francisco, and maybe here too. Wonderful farm. The input of the farm is ground up trees, sawdust, which they get for free. They inoculate it with a wood fungus after they sterilize the sawdust, and they grow things. They put it in bags and add the inoculum and rack it, and they start to grow the fruiting bodies of wood fungus, which are delicious. They harvest them, they sort them, and they bring it to market. Huh. I can take a ground-up tree and make something. The guy who brought me there turns out to be an artist, Phil Ross, and he was doing things like growing bricks and making tea houses and putting them in museums, or growing chairs, and so on. Well, maybe I could get really good at moving my computers into wood fungus and programming the patterns they grow into so that I could make particular patterns, like what I want. Maybe the pattern that is inside my cell phone. Could we begin to think about doing that? This is a computer simulation from Radhika Nagpal showing the programmed pattern formation that she's coded into a reproducing cell in a simulation. We haven't done this yet inside actual cells. But what if I could do this inside a wood fungus? What if I could combine that with the capacity for biology to make hard things, so-called hybrid materials, like the sponges that grow in the ocean and make silica? Right? These are examples from Joanna Eisenberg looking at the silica skeleton of the Venus flower basket sponge. 10 centimeters. Grows itself under environmental conditions. You don't have to heat it up to 1,500 Celsius and have a huge energy load and so on. What if I could actually take a wood fungus, ground up sawdust, throw in some minerals, some crushed oyster shells, and let that compile for 72 hours, and I actually pull my cell phone out? That sounds like a type of manufacturing I could get into. What if we push this further? What if we, see, we're not changing ourselves, we're changing how we exist, right, in a way that has a chance of being better harmonized. A couple more examples just to poke you a little bit, I hope. Sissel is a smell artist in Berlin. We put her in a microbiology lab at Harvard. What would the artist and the microbiologist do? Well, Sissel turned out to be super excited about cheese and the smells of cheese. And the microbiologists, well, they like cheeses too because they have microbes inside them. And they went to the cheese shop and they got stinky cheese and they started plating it out to look at the microbes that make the different cheeses. But pretty quickly they realized that some of these artisanal cheeses are made in small batches by people. And people, we have microbes on us. We're just really starting to understand this. What's the relationship, they wondered, between the human microbiome and the cheese microbes? A classical approach would be to take everything apart, sequence everything, compare it. A synthetic approach is very different. You would go around the Harvard campus and you would collect microbe samples from your colleagues. You would go to the farm and you would get raw milk. You would inoculate it and you would make cheese. You would synthesize cheese. Well, it turns out cheese made from the microbes found in Daisy's armpit is beautiful. It's a citrus floral bouquet. As you might expect, cheese made from the microbes collected from a philosopher's toe is wrong. You need to invent a new adjective to describe how foul it is, and so on. I shared this experience with a banker at Fidelity, thinking I would disrupt his meal. We were having dinner after I first saw this a couple years ago. And he had a very different reaction. His reaction immediately was, billion-dollar market. Celebrity cheese. Okay, so let me wrap up by giving you a stretching quiz, I hope. Take all the things I've shared and let's imagine something. Oh yeah, I should ask. You know, when my mom said, eat my broccoli, you know, you are what you eat, this project is interesting to me. It pokes through that and reverses it. It says we eat what we are, right? It forces a reconsideration of the boundary between biology and technology in a strange way. You might see in the news recently, Michael Pollan, who thinks a lot about food, has been caught up in this project, and he's made some cheese from his belly button microbes. Well, okay, here's the put it all together from a technology perspective. This is a little practice undergraduate puzzle. So let's say you give the folks here enough standard biological parts that you could reprogram E. coli, which normally smells like indole, which stinks. You get rid of that, and instead the E. coli make methyl salicylate, which smells like wintergreen, or isoamyl acetate, which smells like bananas. They did it. They called their project Eau de coli. All right? How does this project impact this gentleman? Well, think about the following. Instead of having centralized manufacturing from plant and petrochemicals that goes into a perfume, that goes into a bottle, that's shipped around, and people slather it, instead we have centralized integration of DNA sequence information by empowered research teams. That data, that sequence information, goes onto the internet, goes onto iTunes. Apple sells it to people for 99 cents. You download the sequence information. You print the DNA encoding that information. Oh, that sequence information codes for the enzymes that makes the odorants, the perfume molecules. Given that DNA, which you make locally, you transform your epidermal ecosystems. You now have living, programmable scents and fragrances. You've eliminated the shipping cost. You've eliminated the fossil inputs. You've changed the material supply chain, right? That's what happens when you begin to recombine Technologies like reuse of parts, synthesis of DNA, distribution of information over the networks, and biological manufacturing. I don't know that it's a great example. We would certainly still need Mr. Pitt to market the product, and we'd have to address many, many other concerns around safety, acceptance, and so on. In thinking about advancing our knowledge of life and our capacity to engineer biology, I think it's important to say out loud, my hope is we don't replace the jungle. My hope is that we figure out how to partner with it better to make the things we need. As we do this, and it'll be a long struggle, we'll come to understand life, learning by building. If we pull it off, if we enable all of constructive biotechnology, we have a chance of renewing humanity in partnership with nature. If you think about it, we've got 24% of the land left on the planet, which isn't under human cultivation. We are estimated to lose 100,000 species a year. Loss of biodiversity. We have to figure out a different way to operate. And I think in partnership with biology, not to change ourselves, but to change how we make and use the things we need, is a chance forward. Thank you again very much. Sorry to go a little bit over. Happy to take questions.